So thank you very much for inviting me for this conference. It's impressive to be in front of such a large audience, even if I don't see it directly. Well, my first reaction when Pauline proposed me, she proposed me this exact title, The Manager's Expectations of Model for Health Issues at the Wildlife Livestock Interface. And when seeing this title, I said, oh, Pauline is considering me as a manager. And I'm not so sure of that, but there are also other people considering me as a modeler. And so I hope that the reason why inviting me was to make the link between the two. And so I don't know everything about management, but I don't know everything about models. Most of all, I think that today, for example, I have seen titles. We'll learn very interesting things after my talk. But I try to make a panorama of some ideas I got from my work, primarily presently in ungulates, but also in carnivores and a bit more anciently in cats in particular. So, well, what's the issue with interface? We are talking about interface very often now because it's the place where contacts occur between sources and targets of diseases, and particularly we think about emerging diseases. And the reason why we are talking more and more often of interface is that the interface is growing more or less. We have here a few illustrations proposed by Serge Morand about the increase of the domestic populations. And here I focused on the interface between wildlife and domestic animals. You have a lot of literature on the interface with humans, but I try to just focus on animals because it's well, it's enough for me, more or less. So you see that here you have the example of the total head of cattle around the world. It's growing very sharply. The rate of contact increase, which was here estimated by the number of reported threatened animal species. So this way of reporting can be discussed, but it's an indicator of the fact that the rate of contact with these species probably increases. And of course, the rate of emergence increases, even if here also there are several causes for that. So it's a growing problem. And what happens at the interface in terms of epidemiology? Well, the central issue is the infectious contact between two receptive species. And this is really the point I want to focus on. So we are in between two populations, and one is the source, and most often the source is considered as the wildlife population because we are focusing on a question of wildlife to domestic transmission. But you can also consider that the source is the domestic population. And starting from these infectious contacts, you can have several results. One is just a spillover, so just a small amount of transmissions. I try to see if I can see the chat while presented, but I don't think so. I don't see it. Do you have an idea?
No, but I can see the chat, so
okay.
there is no question.
So, well, anyway, I didn't tell that, but I try to keep some time at the end for questions, so I hope we can do that. So starting from these infectious contacts, you can have either just spillover, local outbreaks. So I will talk especially about brucellosis in ibex because I studied that, but you can think about avian influenza, for example. And you can have, if the pathogen is able to share to be transmitted in the target population, you can have even epidemics, pandemics, shared diseases, endemic situation. And I also considered the case of complex life cycle parasites because they are crossing this interface in all their cycles. And I think they are good models of transmission at the interface. So there are a lot of literature on the impact of source population abundance, structure, and disease dynamics within this population, on the ability of pathogen to cross barrier because they have, for example, viruses, the ability of have a high number of mutation and so on. I will focus more on the epidemiological part of the contact, which is really the contact pattern. So I looked for a definition of interface, and in terms of epidemiology, I was not able to find really a good one. Even sometimes in titles of papers, there is a term interface, but there is no definition in there. And possibly there is one somewhere, but I didn't find it. So I found the definition in terms of physics of a shared boundary between two system components. So we could represent it as just a line between domestic and wildlife. And in terms of model, it could be easy just to add a term saying that there are contacts between infected individuals from the source and susceptible individuals from the target population. Now to go further, I try to make a small outline of my presentation. I will first show that we are dealing with a structured problem, a much structured problem, a multi-population issue, an asymmetric problem, and a multi-actor situation. And at the end, I will try to define what should be a kind of ideal model in this context. So when looking at the precise mechanisms at the interface, the mechanisms are going either directly from the source to the target population. So what happens there, it's an issue of pathogen emission, pathogen excretion. So it's an issue of how the pathogen is spreading within the host, its pathogenesis, microbiology, and so on. And it's a matter of how the pathogen is spread by the individuals of this source population. So it's a matter of a lot of behavior. On the other side, you also have a question of behavior because the exposure of the target population is also related to behavior. And in the middle, you have the survival of the pathogen in the environment, the structure of the environment, the landscape, the fences, and so on. And you may have vector dynamics and behavior, but it will depend on the transmission of the disease. And last, in the target population, you have the question of receptivity. And I will not develop this point because it will be long this morning, but you can also take into account the fact that, for example, at the interface, the immunity of the target population has to be good. And if for some reason the individuals that are at the interface are not so able to fight the pathogen, that it can be also a condition for emergence. And it can be something that is useful to consider because the interface is not necessarily the normal area where this target population is living. And for example, you can have some degraded environments, and the interface is there in these areas where the immunity can be maybe not optimal. So when going to biologists, the modelers will ask questions often, and about population biology of all the species involved, about pathogen transmission routes, about virulence, and so on. And just about this interface, they will need to ask questions about excretion pattern of the pathogen. And here, for example, it's a question that is not so well known. And for example, we had to deal in France with the question of whether fox, red fox can be a host for bovine TB in France. And so we ask, well, how does the fox is going to excrete the bacteria? And in fact, it was not so easy to find this information. We had to perform some experiments in France to determine that. Then you ask for the dynamics of the pathogen in the environment, the survival and dispersal. And again, to take the same example of bovine TB, the survival of mycobacteria, it was known for long that it's surviving in the environment, but the variation of this survival and so on were not really very well known until the moment that the question of the interface raised this question again, and new studies began to be done. And this is the same for avian influenza virus survival in the environment. Before we had this interface developing, we did not really know how is this virus surviving. And the third point is, how do the contacts occur between wild and domestic animals in this particular environment that is the interface? And interface is not the wild environment, it's not the domestic environment, it's in between, so it's a particular area. So the first question is which contact? Something that I don't agree so much is that often in field studies and field studies that are used for models, people consider that being at the same place and at the same time, we are more or less at the same place and the same time, so less than, I don't know, 1 meter, 10 meters, 100 meters, means that there is direct contact. Because the data are not going further than that. And they also assume that if you have two animals at the same place but different time, you can have a possibility of an indirect contact. Well, it's maybe a more complicated than that, because you have to detail which type of contact really is occurring, and does it correspond to the expression pattern. Here on the right, you have a young male ibex with goats, and can it be a problem for brucellosis transmission? Well, as long as they are not mating, there is absolutely no risk in this situation. They are clearly contacting, but the only way for this male to transmit to goat is mating. So if you let them together, they are going to mate, but if they don't, there is absolutely no risk. Then you have the question of, and this is because for the male, the excretion is not by, for example, saliva or urine. It's only in the sperm, and so you don't have anything else than mating for a male to transmit. You have the question of intensity of contact and what is the probability that a contact results in a transmission. And here on the left, you can see that the contact, the cow is licking. I like this picture. I don't know where they have taken it, but it was published in a veterinary journal recently in France. And this is different from just being together 1 meter away from each other. And the third point is, what is the duration? And for example, when you say there is 1 contact per day, what you have on the right here, it doesn't account for the fact that the contact is lasting 1 day long sometimes, and sometimes it's just 1 hour long. So here we are entering in which I would call a kind of behavioral ecology, but it's interspecies behavioral ecology. So just to show what can be done and what cannot be done in that area. The point is that behavioral determinants of these interspecies contact, and for example, attractivity or avoidance, are not in the core activity of ethology, even if it is growing. So in the normal ethogram of a species, the question is, how do the animals interact with each other within the same species? But contact with other species is not a question that is usually raised. And so you need to have specific data, and specific data on these contacts. Here, so the contacts, what can they be? I try to think about the longest possible contacts. And in terms of direct contacts, it can be, I think that the longest would be mating. You can count it in minutes. And all other contacts would be either in seconds or even even more rapid than that. If you think about aggressive encounter or predation, it's very short contact. And so the problem is how to measure that and how to observe that. It's very difficult. And for indirect contact, here you have some extracts of videos from the thesis of Ariane Payne. You can see badgers in both pictures and one cow in the upper one. And I don't know if you can see exactly what they are doing, but I think it's very difficult to see. Are they really close to each other, or will they be close in the moment? Are they just sniffing the food, or are they really eating, which is different in terms of capacity of transmission? So you have short sequences, cryptic behavior, and so it's always difficult to observe. So if you need some data on contacts, you must know that you have some hypotheses to do, because it's very difficult to observe. There is a recent review on this direct data about contacts, and you see that the possible methods are quite a lot, which is nice, but not all species were studied with all methods. And for example, you can see the wild horses. Wild horses were observed most by GPS, and other species were observed more by other methods, like for example, on the upper part you have sheep, goats. So it's the species like chamois and ibex. So it's in the alpine pasture, and most of them were observed by direct visualization. So it's completely different from GPS data, and probably all of them have limits, not all the same limits. A general limit is that if you are interested in small species, in small birds, in small rodents, more or less they are never observed. So you don't have any real observational data. If you need some information about the behavior, and you need videos, but again, you need to have the video at a very precise moment, and when it's by night, it's very difficult to see. And the GPS precision itself is often a bit limited. So just to illustrate that different methods may raise different results, here we have a result of a comparison between methods to assess the interface between bush pig and domestic pigs in Africa, in Uganda. And you have the results of the camera trap survey, of track survey. So the track survey is the one with the very little tracks on each circle. And you have a questionnaire. And no, sorry, the tracks are for the questionnaire, and the circles are for the observed tracks. And you see that if all methods were giving the same result, you should have dark tracks within black circles. And it's not exactly like that. So each method is obtaining a bit different results on the same places. And it means that, and here, for example, only camera trap allowed us to identify factors that make these contacts vary between places. So each method has different bias. And so if you take results from a single method, you can have some inappropriate conclusion and recommendation. And the idea is ideally to combine several methods. So most information is issued from observation. And we understand that because it's very useful, but we also have some experimental data, and it's interesting to see that I think that without these experimental data, we don't have the same level of information. One I like very much is an old study in New Zealand about cattle that were shown with sedated possum. So the possum were just left sedated on the pasture, and they observed interaction that were completely different. So it was to mimic sick possums on the roaming on the pasture, and they obtained quite interesting result with cattle following and licking and sniffing animals. And they show also that if you have dead possum on the pasture, it's completely different. So you cannot just have information from dead animals. when you have this information, what to do with that? Well, remember that transmission dynamics will be depend on this contact, and not only on the average contact rate, but really on the contact distribution. Of course, this is probably not a surprise to you that the contact distribution is quite skewed. And here you have a small example about the visit of latrine by badgers. And you see that most individuals do not visit anything, but you have a few individuals visiting a lot. And this contact distribution combines with other levels of heterogeneity in the rate of contact itself, in the type of contacts, in the intensity and duration. And also you have the strong heterogeneity, which is much known in the level of excretion and probably in the level of susceptibility. So if you combine everything, you can have very different situation. For example, if you have very long contacts, you can have a big R0 value, but a long duration of contact means that you can have a few cases, but then the disease and the outbreak may get extinct if there are only few long cases. And on the other side, you can have very few contacts, but if these are high probability contacts, are not maybe smaller than one, but if a few contacts are possible and they have a high probability of transmission, the disease then may persist a long time. So it depends on especially what happens to these individuals in the blue area. And if they are, for example, very specific individuals that have very, I don't know, aggressive behavior or a specific excretion pattern of these individuals are in some way particular, then a few contacts from them can be much more important than hundreds of contacts from any individual in the population. So to illustrate the impact of this heterogeneity, I will show a few information about the brucellosis in ibex, because these last years we were a bit busy about that, because this area of the Bargy Massif is the only wild ungulate population that is persistently infected in Europe. So it started in 2012 with first cases discovered first in people and then in the cattle, and then in ibex. And so monitoring began, and first observations showed that old males were the category that were most often seropositive. And so at the beginning of the story, massive culling were done, and they focused especially on these old males. And then after a few years, we were able to, with the information that was available, we were able to develop an individual-based model representing the situation of the massive and in particular the spatialization of the disease. And this model first showed that individuals have a heterogeneous contribution to transmission. This is also probably not a surprise for you, but we can remind it quite often. And it's mostly revealed that females, in fact, are the source of the vast majority of transmission, both within spatial units, so nearly all transmission, but even within even between spatial units also, because males are transmitting infection from one area to the other, but males are always infected by females. And so the females are the source of most cases, and males are not quantitatively so important. And so probably this is a reason why the first culling operations were not so efficient in lowering the force of infection, because it targeted males, and probably it could have been more interesting to target females. And including for cattle and sheep, when the farmers say, I see male ibex, because male ibex are quite bold and they come often in contact with a goat goat and sheep. And the farmers see males much more often than females, and they worry about that. But probably if we are not in the reproduction season, males are not really a problem for the interface. So to illustrate this temporal, spatio-temporal heterogeneity, well, we may think about seasonality in behavior, seasonality of pathogen survival, daily pattern of behavior, and this habitat selection that can be very specific. So here you have the result of observation of the location of indirect contact in alpine pastures. And you can see that the stars are the contacts that were observed, and the gray areas of specific areas. So it's generally this specific area are areas where salt is deposited by the farmers to feed the sheep with salt. And you can see that, for example, in the second drawing here, all the contacts that were observed between chamois and cattle were all situated on this salt deposit. And another possibility is a wildlife refuge, but in all cases, a great majority of contacts were on these specific areas and not everywhere in the pasture. So it's very heterogeneous in terms of very, very local area. It's also heterogeneous at a more large scale of landscape. To illustrate this is a model of toxoplasmosis in farms. So here the contact between domestic and wildlife is the predation by cats on rodents. And here we model the resulting level of environmental contamination, that is the source of contamination for domestic species and for people. And on the left, you have the observed situation. It's a village of northeastern France. And so in these villages, everything is clustered and the farms are within the village. And you see that the environmental contamination is very high within the village, but very low everywhere else. And so here is the place where people have their gardens, living outside, the children outside, and so on. And so probably the risk is very important here. And if the farms were located, it's a simulation here, if the farms were locating a bit everywhere in the landscape, it would be different in terms of risk, because these hotspots of environmental contamination are depending on how the landscape is organized by human activity. And to go a bit further in terms of spatial heterogeneity, we have the urban-rural gradient. And here again, we can think about how the population of wild species are reacting to anthropization and urbanization. Here you have the model simple description of this gradient of urbanization between forest and central urban areas. And you can see that more or less cat density is following human density, but rodent density is not. And so the level of predation is dependent on this landscape because of relative density of species and because also of anthropogenic food supply for cats. And this translates into different dynamics of the toxoplasma cycle, because toxoplasma cycle is formed by two types of cycle. One is a cat-rodent cycle. So it's the majority of cases, majority of transmission in the rural areas are this complex cat-rodent cycle. But what we show is that in the urban areas where there are not so many rodents, there are a lot of anthropogenic food supply. And we could think that maybe toxoplasmosis is not really a problem. In fact, you can have a relatively high cat environment direct cycle. And so you have or not of toxoplasma that is composed differently depending on the place on this gradient. So to close on this heterogeneity part, I try to outline the perspective, and I think that for the moment there are very few models integrating direct contact into models to link this contact observation and what happens at the epidemiological level and the consequences that can be observed at the indirect data. Here you have a recent example of data that are directly used within a network model of badgers, latrines, and cattle. And it's interesting to show that it gave some new results on the role of individual badgers, which is correlated to their role in the role of badgers within the badger network is correlated to their role in the overall network, which shows that probably the individual characteristics that are important within badger transmission are also the ones that are important at the interface. So for this first aspect, which probably is the longest one, the interface is structured temporally, spatially, demographically, and behaviorally. So I represent it as a line, but no longer a straight line. And a few other observations. First, going within the thickness, what I call the thickness is what is exactly within the interface, and what we call wide. So wide is a context-dependent definition. For the law, generally wild is a list of species. But wild is also what is not under human supervision. So in all cases, it depends on whether humans have selected the animals and humans are controlling the animals. But it can be somewhat different depending on what you consider and depending on the, for example, the new European Animal Health Law considers feral domestic animals in the wildlife compartment. So I try to make an inventory of what can happen within the interface between the own domestic species on the left and free wildlife on the right. And if you think that all situations can occur with domestic species in blue. So for example, extensively farmed domestic animals are clearly within the interface because they are in contact with wildlife. You have free living species that are in black, and you have these populations with a variable status because feral population of domestic species may be considered as the domestic or wild depending on the law you are considering. And you see that, in fact, there are many different kinds of populations that can be involved in this interface. And a few of them have been most considered recently. For example, this commensal wildlife, so the idea of bridge species between avian farms and wild bird, for example, the game farming and everything relating to game enclosure and game farming for different purposes. But there are also different populations. For example, you can even think about wildlife in rescue centers getting in contact also with domestic and humans. So for the moment, I think that there are not so much information on the role of these populations. They may have different statuses in terms of whether they are spillover hosts, whether they are maintenance hosts, and how the reservoir we are looking for very often is constituted. And for example, what is the role of the so-called bridge species, which could be a role of going from the reservoir to the target host. For the moment, there are very few models on that. Also, there are lots of questions. And to come back to the example of ibex, in fact, we really don't know how the infection crossed the border from ibex to cattle in a few years ago. And so we think about maybe dogs bringing something infected to the farm, but really we don't have the information here. for example, to illustrate that infected populations are not necessarily reservoir. You can think about any population where diseases is not really persisting. Here, a model that was drawn from for African horse sickness in zebras. And the question was whether zebras can be reservoir for local horses. And you see that in this situation, so it's not the same in all populations of zebras, but in this situation, you have a small population of zebra in the Western Cape province. All populations are quite small. And so the population is not able to sustain transmission of this African horse sickness. So probably they are unlikely to represent a long-term source and to be a reservoir of infection for the local horses, even if you can find some infected individuals. So here the perspective in my idea would be to model the role of this breed species. I don't really like the term of breed species because I would prefer to use the term of breed populations. But we can have breed populations of commensal wildlife, and I think that maybe there are some models about avian influenza virus. I didn't find so many models, so I have a one here, which is a model, basically, because it's a, a drawing reminding the role of pigs in the transmission of Nipah virus from bats to humans in Malaysia. So here we have a case of a virus able to be transmitted by a breed species in some circumstances, and without breed species in Bangladesh, because there are other ways of other trees, other ways for people of using these trees. So I think that it can be useful to think about these small populations that often maybe modulus, we tend to simplify the system. So to skip this kind of few individuals going in between the two populations, but they can be important. And I think about, yes, bridge populations, farmed wildlife, and even others like traffic and exotic pets. So the interface is structured and what I would call thick, because there are many things in there and even different kinds of population within the line. A third point, going for more information on maybe a human side of the interface, is that it's basically an asymmetric problem. To illustrate that, I give the example of the pestivirus. We worked with Gaël and Pauline. And the reason for studying this virus is that it causes a lot of major outbreaks in Pyrenean chamois. There was a lot of mortality, and the managers pointed that the risk is that sheep transmit the virus to Pyrenean chamois, and this is the cause of the population decline in the Pyrenees. So here the manager was the hunters, and they were clearly accusing sheep farmers for transmitting this virus. So Gaël wrote a model of within chamois model transmission, and this showed that you can see that actually there is increase of population size, and when the virus is has entered in the population, there is a sharp decrease due to the virus. And the model showed that the mortality of infected individual is really high. So it's more than half of individuals dying in a few weeks. And that the dynamics depends on persistently infected lambs. So persistently infected individuals are really the key to this epidemic. And so when you think about what is the risk of transmission at the interface, you can see that here you have the reproductive cycle of chamois and sheep. And you see that for the chamois, the mating is in December. So to obtain a persistently infected sheep, you have an at-risk period that is going from December to February. And at this period of the year, there is absolutely no contact between sheep and chamois. And so it means that probably to infect chamois, it's quite difficult because susceptible chamois, susceptible pregnant females are not in contact with sheep. But for the other way, you can breed sheep with two different kinds of mating. You have autumn mating and you have spring mating. And you can see that with autumn mating, the at-risk period is in autumn, so there is no contact with chamois. But if you go with spring mating, you have the at-risk period that is covering the period of contact on pasture. And probably it means that the transmission from chamois to sheep is much more likely in this case than sheep to chamois transmission. So this was a case of really asymmetric transmission. And this is also asymmetric because of our point of view. Most often we are on the side of domestic activity, and the questions are raised by managers are often raised by managers of domestic species fearing transmission from wildlife. So we think about this domestic side like something that is relatively stable, relatively controllable, well known and at least familiar, and more easy to control. And if we think about wildlife, well, it's environmental variation, not so controllable, not so observable, with structural uncertainty. And so our point of view is not for everybody, but it's easy to consider wildlife as the potential reservoir and even the ideal culprit sometimes. So it can be in both in both ways, but I think that most often it's this one. So the perspective here would be to develop symmetric questioning. And beside, well, the left one is the classical one. For example, you need to protect livestock from wildlife reservoir. You need to design efficient biosecurity. And so you need to study the behavior of excreting wildlife and of susceptible domestic targets. The other questions would be how to protect wildlife from domestic reservoir, but also how to avoid the emergence of new wildlife reservoir, and then to protect livestock from spillback. So the aim would be to design also biosecurity measures, but other biosecurity measures, like, for example, how can cattle transmit TB to badgers on pastures? And you have different questions about cattle excretion and badger behavior. So it's just a different way of seeing, but probably it also has some interest for wildlife and for domestic species also at the long term. And so now the interface is structured and it's thick and it's also asymmetric. And the last point is that it involves also a lot of actors and an evolving situation. So on both sides of the interface, you have managers. So you don't have a single one probably, and you probably have many of them on both sides, each with its own piece of information, each with its own vision and aims, and often not discussing and sharing them. And the situation can evolve. In particular, the situation can evolve because you are applying management measures. So I will not develop that, but you know that a long-term effect of management in wildlife can be summarized by sometimes a kind of perturbation effect, which occurs on the managed population. So for example, you have impact of density on density-dependent population dynamics. This has been showed for various species, but I think about wild boar, for example, which is much reacting to management measures. You have the impact on spatial behavior of hosts, for example, in badgers in culling areas. And you have effects on other species through the disturbance of species, fences limiting dispersal, and effect on predators or prey of the removed individuals. And so these changes in the situation tend to complicate the relationship between actors even more, because these decisions of management have been taken by some managers, and so others can be angry because of the consequences. So a small example here of a very simple model we did on FELV virus in domestic and wild cats. So the idea was just to have two populations, and here the contact is dispersal of individuals between the two populations because they can mix and produce hybrids between the two species. And here we just modeled the fact that FELV is a virus with a strong impact on population dynamics that can be managed by vaccination and elimination. And the idea was, what happens if you manage FELV in domestic populations here that are big populations, because the abundance is quite important. And what happens in the small population of wild cats that is in contact with these domestic cats. And the result was that management, and imperfect management in particular in the domestic population, is able to increase disease persistence in the wild population, because the management leads to an increase of the domestic cat population dynamics, and so increase of the flow of dispersal to the wildlife. And so the persistence of the virus in the wild population is increased by the management in the domestic one. To illustrate that in the case of brucellosis and the actors problem, so I said that in 2012 we had some cases in children, in fact. So this was a problem of zoonosis and of the Ministry of Health. Then we discovered cases in cows, and so it was a problem of agriculture. And here there is an area of production of a very important cheese for French gastronomy, so it's a very big issue. And so the first idea was to manage the situation in cattle, but farmers are not very happy with that because you have to cull the whole herd of cattle. Then they discovered these cases in ibex, which is a protected species. So the Ministry of Environment was involved in that. They discovered cases also in chamois, which is a hunted species. And so here there were a lot of conflicts began to emerge with hunters, association of environmental protection and agriculture. And the last point was that it was an unexpected situation. So a lot of factors came from research and expertise. And so this multiplied the number of people dealing with this situation. Then you can add also politicians, medias, and also other people working in other protected area and wondering what is going to happen in their area and what is going to happen on their heads, more or less. And you can see that with time going on, the conflicts were very possible between these so many different actors, and they are still going on. So the perspective here is to develop models that will include indirect and long-term effect of management. And in this, I include a lot of things that we added from time we are going on from time we are going to work on this ibex system. So usually models predict the epidemiological outcome of management in terms of extinction, prevalence, risk, and economical issues often. But they could also consider more often the perturbation effect of management, so the indirect impact on population dynamics and so on. The conservation issue, if there is a possibility that the management is going to impact the conservation of the species, the impact on other species that are present in the environment. For example, the impact of fences in a protected area can be devastating. But also the financial and also the operational cost, the feasibility, the risk for the operators that are going, for example, in the mountain of Bargy to capture ibex. And so there is a limit to that because it's an area where you cannot go without a risk. include also some ethical consideration. I think that it's not only associations of nature conservation that should have these ideas in mind of how many individuals should be removed and how and which one, and consider the acceptability, the risk of even public disturbance of going to judicialization of the problem. So to end with this example of Bargi, I show you the evolution of the epidemiological situation. These last years, the program of control was mainly both based on test and cull, because culling non-tested ibex is not accepted by the associations, more or less. And so prevalence declined, and now infection remains present, and it's even locally quite active in the central area of the massif. So now we try to use models to predict the possible outcomes in the future years. So here you have two scenarios of what happens every year and what could happen in terms of incidents in the area. And we try to model what happens in terms of epidemiology, but also to answer the questions that we have about ibex conservation, ethical consideration, the cost, the risk, and the acceptability. And we try to use that to communicate with different actors. So next week we have a meeting with the general local sanitary authority, National Council for Nature Protection, Ministries of Agriculture and Environment. So we try to go between these different people, and I must say that we did not obtain any general agreement for the moment. And so now interface is so structure, think, asymmetric, and multi-actor. And my last point was what to do with modeling. So you know that modeling is an interdisciplinary challenge. So this means interacting with a lot of people around you, learning from people for example, pathologists, ecologists, or microbiologists, for those of you who are mathematicians. I would like to insist on the fact it's also teaching these people and allowing them to use the models so that you can interact in both directions. And when going to managers, the idea would be to interact with all managers, so not only one, because probably at the interface you have several managers, and interacting with them since the beginning, because if you go to them at the end with your model, probably it will not be exactly what they need or what they want. So I let you guess here who is the manager and who is the modeler, but the idea is to ask, what do they ask for? And they can ask for a relevant solution, but they also can ask for a better understanding, or something, a tool to communicate a bit with other people. What do you think they need? That can be quite different. And also, what do you want yourself as a modeler to do with that? And so the idea is try to find a way of agreeing on what we can obtain together. So learning from managers is important, because asking the relevant questions, they have often a knowledge of the system, or at least part of the system. And even if you ask relevant questions, they can improve their biological knowledge. And I think the other point of teaching to managers is also very important, because they need to be able to use even the models themselves. So if you use very simple models, develop real-time models, ready-to-use models, open models, models that will still be available after the student has gone, for example, models that can be really used by people themselves. It can help a lot, and I think we are not completely at this point for the moment. And so to conclude shortly, the interface. So the interface is like that. So it's structurally and it's structured and thick and asymmetric and multi-actors. And so probably we need disease models, disease-specific models and species-specific models. Not only that, but in particular that ones that are structured, that consider all the populations and also all the questions, even if sometimes a question is not raised by the manager, maybe it can be useful to raise the symmetric question about the risk, for example, for wildlife for later. And possibly models that are able to make managers communicate and to be able to be upgraded, and this within a long-term and open process of modeling. So this is not what I do, but I think I should try to do more of that. Thank you for your attention, and I want to thank many people, Pauline and Elisabetta, of course, but a lot of people, some of them are clearly managers and some of them are clearly modelers. So we try to stay in the middle. It's a bit long, this. Thank you.
Thank you, Manuel. It was really a great talk and I learned a lot listening to you, and you answered almost all the questions I had. So thanks a lot. We had a few questions. We have not so much time for questions, but we can still take a few minutes. And there were two questions at the start of your talk about the definition of interface and about well, I will let the people ask that question. So Adrienne Ardeline ask a question, if you want to explain it orally. And also Matt de Jong, you ask a question.
So the one about observation, the definition of interface to be included in future revision of the OIE standards. Well, I don't know what are the OIE standards about the definition of interface, in fact. So yes, maybe if they have a definition and we can improve it, it would be great. So the idea of the paper we are going to write then, yes, is to redefine interface as a more complex idea than is just thought. But I don't know if they have a definition, in fact, of OIE. I don't know if somebody has the information. I'm interested.
At this time, I don't think the definition exists, but we can ask to be included for the future revision for the next year.
Okay.
Thank you.
So Matt, you ask a question. Do you want to?
Yes, so thank you very much. It was very, very, very good presentation. Very nice, very complete. But I think that maybe one thing should be added, and that is that the pathogens may be different in different species, and especially, of course, in the influenza, we know that viruses have gone from humans to pigs and then became adapted to pigs and not easily go back to humans again unless they acquire genes from avian influenza. And so there's also this complex interaction where the pathogen is not the same. So some are really clearly multi-host pathogens that have their cycle in the vector and in the mammalian host, but there are also that really adapt to the different and in between. So but this adaptation of the pattern, I mean, working in the influenza, that's a very important. But I liked your presentation very much. Thank you very much for that. Really good to have such an overview, and also to emphasize the back and forth between the wildlife and the domestic is, I think, an important thing to realize. Yeah, thank you.
Thank you very much. Yes, I know that there is this evolutionary component and adaptation component of interface. Well, yes, I definitely have to add this, even if I could not develop that, because it's even another subject.
It's really nice to have such a complete overview, so that I thought, okay, maybe that's the point to have somewhere in the discussion or so. If you write it up. Yeah, nice, very nice.
I have also a question. Maybe you can share a bit more your experience on from your experience. Did managers change their view or progress in their knowledge about other points or other priority by co-building models with academics and then at the end using the results or even using the models, or finally does it contribute or does it doesn't change anything?
Yes, they change their mind, but sometimes in the unexpected way. Because it's really funny. This year we are going to go back to the field to interview farmers, for example, and it's very surprising to see that some of them are a very different way of thinking that we thought about. Sometimes they think that the management measures were not sufficient. Sometimes they think that it was too much. So they didn't see the models themselves, but I think that first they have very much more varied way of thinking that we thought before, because we thought that, for example, farmers all think the same way, but they're absolutely not. And then seeing the models, I think that yes, it can change their mind, but only if they really have the impression to understand what happens and not showing just the result of like a magic box. And that's the reason why very simple models are very difficult, but sometimes you may have some different versions of the model and be able to give something very simple with it's not to say that it's difficult to give something more, but often you have not so much time to explain and so on. And so giving something a bit more simple can be very useful. If you think about this example of males versus females, for example, I think that in that way they did change their minds. The people that are capturing ibex realize that females are much more important than males. This is a big problem for them because killing a male for hunters and field people is not much a problem. Killing a female is really a big problem because it's the future of the population, you know, and so on. So it's much more difficult, but they understand now that it's better to have few females now, and so be able to let them grow after. Well, I'm not sure it's only the model itself. It's all the information we have discussed also.
Let's see other. Oh yeah, Claire has a question. Claire, you can just take the mic.
Yeah, hi, Emmanuel. Can you hear me?
Yes.
Yes.
Hello.
So thanks for this great talk. My question is more related to wild animals that can cross borders. And I just wonder if you have ever collaborated with neighboring countries in order to, for example, I don't know, design a common disease surveillance systems or population surveillance systems. And if yes, what are the challenges you faced when you had to collaborate with these countries?
Regulations.
Like, I don't know, between Spain and France, when you look at the different wild animals?
Yes, we work with Spanish colleagues and we didn't really try to imagine common ways of managing, for example, pestivirus in the Pyrenees, because the situation, the biological situation is not so comparable. They have different problems, different population, different viruses, it seems, also in France and in Spain. But I think that the most important is that the regulation is completely different. The way of organizing hunting, for example, is completely different. I think that a more maybe a closer relationship can be found in Italy with the ibex management, which is closer, but we didn't really try to find something common because we only have this very particular population of Bargi. So the Italian guys do not try to find any management measure. Yes, I understand that finding common management on both sides of the border is something that probably is particularly complex. And no, I don't have any particular experience, but I'm not sure I want to.